Okay, hello, good evening, everybody. Welcome to session 7 for the course on advanced concepts in cancer biology and precision medicine. We have a few people joining in later as well. I'll just give a brief overview of what we have been discussing over the past couple of weeks, and then I'll revise what we had discussed in the last class and move on to the discussion for today. Again, just to reiterate, people who are not familiar with the way we usually conduct the classes are that we have a discussion session, and at the end of it, I give some key takeaways and some questions to ponder upon and for you to explore it. What we have been following over the past few weeks is that we have the discussion later in the last session, which is on 31st, and we'll have a overall discussion for all of the questions that have been put forth during the tenure of the course. If you have any questions or any queries at any point of time, you can unmute or raise your hand however you feel comfortable, and you can let me know whatever your doubts are. Just a second. Video is not on. Yeah. You can let me know at any point of time if there are any doubts. And if you cannot see my screen or something, please just let me know about that as well. Connection is not stable, so I'll keep it off for now. Okay. Basically, as part of the flyer, as you already have received, we have been discussing the basic overview of everything you need to know about cancer in the initial few weeks. We discussed the hallmarks of cancer, which are some attributes acquired by cancer cells, and that allows them to become cancerous from their normal condition. Next, we spent a few classes discussing some key signaling pathways that are altered in cancer cells and that allow them to acquire that phenotype that I just mentioned. In the last week, we had started discussions on the traditional techniques of cancer diagnosis and also moved ahead towards some advanced techniques which involved or which were centered around the theme of non-invasive diagnostics. This is the overall pattern we have been following till now. And what we are going to look at today is going to be about exosomes, which are another approach towards non-invasive cancer diagnostics. And I will complete this part, which was left behind, which is the traditional method of cancer treatment. In the next class, we'll be moving ahead towards precision medicine and combinatorial therapy, which has come up in the past few decades. And then we'll move on with the course as the classes progress. Basically, what we had been discussing in the last week was about the main advancement that has happened in the field of cancer diagnostics, which is liquid biopsy. And as the name suggests, liquid biopsy basically means that it involves sampling of some of the liquid or body fluid of the concerned patient, and that is used as the diagnostic that is used for the diagnosis of the patient and to predict whether or not they're going to suffer from the disease, or they are very prone to the disease, or they actually are in which stage of disease progression. Basically, it had all these four domains in which liquid biopsy is applicable. First is screening of cancer, like I said, early cancer detection to predict whether or not they have the possibility of eventually suffering from the disease. Second is to carry out early stage diagnosis so that treatment can be started as quick as possible so as to help the patient recover a lot faster. Um, for early stage cancers, liquid biopsy was also used for prognosis to see whether or not they relapse from the disease and to figure out whether or not they would respond to some sort of additive therapies or adjuvant therapies. Finally, for advanced cancers, the approach is quite different. In that case, the approach is to monitor the disease to see how it is progressing, whether or not they relapse. I had explained what relapse basically means. It means that that same person who had once been diagnosed with the disease and has undergone some sort of treatment, which has led to disease regression, they again suffer from some sort of disease, and in most cases, that disease is a lot more severe because it is usually in a condition where it has spread a lot farther. Let me try switching it on once again. Okay, let's see if it works. This is what we had been discussing. Then this is the definition for it. You can go through it in your own time. Then we looked at the major differences between traditional tissue biopsy versus the approach of liquid biopsy and how it has a lot of advantages. It's not invasive. It takes it's much quicker. It is a much better way of predicting disease. It's easier to avail the sample. However, it also has its own set of challenges, majorly centered around the fact that it does not provide you complete information. Complete information always comes from the actual tissue biopsy because you get the tissue histology, you get to see what cells within that tissue actually look like, and you are able to provide a much more accurate and much stronger diagnosis or much stronger result out of that same test. But obviously, each of these techniques have their own pros and cons. And in the current scenario, both of them are in use. And when I say are in use, I mean in the sense that they are both considered to be equally uh, necessary. However, the stage at which each of them would be used can be quite different. Then we looked at the utility of liquid biopsy, again, early detection and detecting how efficient our therapeutic strategy is. These are the two major ways in which liquid biopsy is extremely promising. What we also looked at was the type of samples which can be used for this purpose, starting off with saliva, which is obviously very easy to procure from a patient, then blood, urine, and the lavage fluid. This was an example of a patient who is undergoing diagnosis for lung cancer. Therefore, the bronchoalveolar lavage fluid is what is suggested here. Depending on the cancer type, the type of fluid which might be more useful can differ. And what you get out of it are different types of information. Like, for example, you can figure out what types of proteins, enzymes, hormones, etc. are present in that individual, whether they have cfDNA, whether they have ctDNA or long non-coding RNA. Basically, different types of nucleic acids can be studied. So this is all of the information you can just get from sampling a patient's body fluid instead of actually uh, subjecting that patient to an invasive surgery, which can have a lot of harmful effects. And apart from this, you can monitor the patient's response and you can check whether or not there is a recurrence of the disease. Then we understood some major advantages, like how initially CTCs were reported, then ctDNA levels were studied, and all of the other progress that happened over time. I'm not going through it because we've discussed this already in the last class. Um, then we looked at ctDNA, CTCs, and exosomes as the three major divisions or three major branches which are extensively used or which are the most promising when you talk about non-invasive cancer diagnosis. So among these, we had looked at ctDNA and CTCs in the last class, and today's focus is going to be on exosomes. So in the last class, we understood what is ctDNA or what is cell-free DNA. I had also asked like students to basically go over this difference between CT and cfDNA. While they are sometimes loosely used, it's not actually accurate. ctDNA is the total DNA, cfDNA is the total cell-free DNA which is present outside of a cell. Among them, the DNA which has only come from tumor is what constitutes ctDNA. So there's a slight but very important difference between them. For us, what is relevant is only to look at the DNA which has been derived from the tumor, because that is what will give you information about the disease, about the cancer. However, as you can understand, blood has a lot of other types of cells as well. Obviously, starting off with your WBCs and platelets and macrophages and all of these. So they would also release their own genomic material, which would be available. Sorry, which would be available. But apart from that, what you have to keep in mind is that RBCs will never contribute to it because they're enucleated. So all of this information we had already discussed, and the what you can derive as a response to that, that we also studied. Like, for example, you can understand what mutations are present in DNA, what sort of epigenetic changes are present in that DNA, whether there is any microsatellite alteration. All of this is understood. Then we understood what is the reason that we are getting these sort of DNA in the blood. So majorly because cells are undergoing um, some sort of a phenomenon whereby they are dying or they are undergoing a process which is leading to the fragmentation of their DNA, and that DNA is getting released into the blood. That is what you are able to capture. So this is what we had discussed. Then we understood how it is actually usable in context of an actual patient who has to be studied. So, like I already mentioned, you can understand what sort of alterations are present in the gene. You can look at signatures, mutational signatures. You can understand how much tumor is present, like how. When I say how much, it basically means that how heavy is the load of tumor within a particular patient. Say, for example, you have two patients, both suffering from lung cancer, somebody who is in an early stage of the disease versus somebody who is in an extremely advanced stage. So, if you take blood samples from both of these patients, you would see that the percentage of tumor DNA, ctDNA, in the blood of the advanced stage patient would be much higher. So that is representing how much of the how much of mutant tumor DNA is present within that particular individual. So these kind of information that can all be retrieved. You don't necessarily need to go into details about that because that involves a lot of next generation sequencing technologies, which is not really our focus. But basically, the applications of getting all of this DNA is what I wanted to like bring to your attention. However, while it all looks very good, you can understand there's definitely some, some challenge or some problem with this, some limitation with these technologies as well. And like you can see here, potential issues and drawbacks that are associated with it. The first factor, the first most important factor is that there is tumor heterogeneity. What does that mean? It means that one tumor within a particular person is never only made up of one type of mutation or one type of alteration. Under normal circumstances, of course, there are exceptions to this. So, if you have 10,000 cells making up a tumor, you can expect that there would be thousands of different types of alterations that could have happened. Now, the problem with that is that from these thousands of alterations, the DNA that you're able to get from a particular patient's blood, which is the ctDNA, that will not actually represent all of these diverse alterations because it's a very limited amount and it's present in very low frequency or very low yield can be actually obtained. So, as a result, you're not getting complete information about the tumor, you're only getting a representation of it. So, this information, while useful, is incomplete. So that's a drawback of this technology. Secondly, for brain associated cancers or for neurological cancers, the tumor DNA which you would be getting that might actually be limited or might not actually be present at all. That is because of the presence of the blood brain barrier. So if you're not familiar with it, it's basically like a mesh of capillaries which is present between the blood that flows into your brain and the rest of the blood circulating in your body. The reason for that is to protect the brain, to protect your neurons from a number of parasites which can otherwise uh, enter the blood flow within the brain, and also to prevent any sort of unwanted movement. It's not just parasite; it's for any molecule which could potentially be harmful for the brain that is protected by these network of capillaries. So they actually form the BBB, which is the blood-brain barrier. Then other problems can also arise, like for example, the tissue of origin might not always be understood, etc. So again, keep in mind, this is a very useful technique and a huge advancement if you think about cancer as a field, but it carries with itself a lot of limitations as well. Then, in clinical oncology, we understood some basic steps that would actually be required to integrate liquid biopsy into clinical practice. You can go through this. It's just majorly just for understanding. Then we understood all of the different types of different regions from where ctDNA can be extracted, which are not derived from the blood of an individual. So, like I already mentioned, the saliva, from bile, from peritoneal fluid, from stool, seminal fluid, uterine lavage, urine, pleural effusion, or cerebrospinal fluid. So that depends on what type of cancer a particular person is expected to have, and depending on that, sampling would also vary. Then, yeah. Then we understood CTCs. This was a sort of timeline from, well, quite early on from 1830 to 2020. So it's a sort of a chronological sequence of events that are landmark events that have been understood in the field of liquid biopsy overall. And again, I had reiterated that you need to understand the seed and soil hypothesis by Paget. It is one of the most fundamental understandings in the field of cancer biology. So it's you need to be familiar with it by now. And as you can see, if you just give this entire picture like a quick glance, the one word that comes up multiple times is metastasis or the metastatic cascade. So why was this important for our discussion? That is because everything that we have been discussing, this whole theme of non-invasive diagnosis, very, very heavily relies on this phenomenon of metastasis. And metastasis is the consequence or is the result of another event which we have discussed very early on in the course, which is epithelial to mesenchymal transition, which is EMT. So when cancer is progressing and some cancer cells or some tumor cells have metastasized from the primary site to other locations within the body, during that process, that is exactly when they actually move into blood. They slough off from the primary tumor and they extravasate. Extravasation, if you don't remember, it was the process where tumor cells were able to sort of pinch off or sort of slough off from the original tumor site. And then they were able to move into the blood, and then they were able to move through vasculature to other parts of the body and then find their desirable new sites and then form new tumors there. So during this entire process, whatever is getting released into the blood is what you're able to actually detect. So the phenomenon of metastasis is something that's very crucial and very relevant to this entire field of non-invasive diagnostics. So among this, you can see all of the different events. They have been characterized, when they were discovered, and what were like who were the scientists who actually contributed to that discovery. All of that information you can obtain from here. Apart from this, one more thing, I had mentioned that we will be discussing exosomes here. So you can, this is also to give you an idea that this discovery has not emerged in one day. It is because of so many scientists. This is just a very small representation of all of the scientists who have contributed to it. But you can see some other events that have also added to this research overall. So just look at it in your own time. I'll share the slides by today or maximum by tomorrow, so you can look at it. Um, yeah, this is the event that I was talking about, that from the primary tumor, there is intravasation and there is extravasation and then formation of new tumors. So during this entire process of intra and extravasation, whatever is getting released into the bloodstream is actually helping us in the diagnosis process. And yeah, these were the different examples of all of the other localizations or the other secondary tumors that could be formed, like lung metastatic nodules, uh, metastases within the liver, within the brain, or within the bone. The most common sites of secondary tumor formation are these. Very commonly, these are the four sites where it actually happens. And why these are the four common sites? The answer again goes back to the seed and soil theory. So just go over it, and it should answer a lot of your questions. Then we had discussed a little bit about the mechanisms which support the survival of CTCs in this early process of metastasis. So CTC survival is very strongly helped by some anti-apoptotic mechanisms, which is centered around the FAS ligand signaling process, and also by TGF-beta signaling as well. So they effectively help EMT, and they also help survival of cells that are a part of that EMT process. And both of these converge to effectively help in the survival and the sustenance of circulating tumor cells. So apart from that, we also understood some points where intervention can actually be made. For example, early cancer detection can be done like when from a tumor. Again, this represents the heterogeneity that I was talking about. You can see different colored cells all forming the complete tumor mass, and from there you can get some early information about cancer. Secondly, you can perform surveillance for micrometastatic disease, that if early cancer detection has been carried out and a patient is found to potentially suffer from the disease in the long run, then there can be constant monitoring of that patient over a period of time, say 6 months or a year, to keep checking for whether there's an increase in the tumor load, a decrease, whether there are new cells with more mutations. They are all indicative of much more aggressive disease. So in that case, treatment strategies could be altered, or basically the disease progression for that patient can be monitored. Then we understood how there can be localization or movement of the tumors to either liver or the lungs, and depending on what stage it is in. And because another thing I mentioned here was that after after they have moved into the liver or into the lungs, these tumor cells can again undergo the EMT process. And from the lungs and from the liver, you can again see some new cells, some new modified, much more uh, much more severe disease causing tumor cells can again get released into the circulation, and then these cells will carry a signature of the tumor site that they have actually come from. So if they're from the liver, they'll carry some signature from there, or if they've come from the lung, they'll carry those signatures. So that can actually help you understand that yes, the disease has progressed much further, and you'll get all of that information, and accordingly the patient will be subject to the required treatment. These were the key takeaways and some questions from the last class, whereby we understood that yes, liquid biopsy is a huge landmark discovery in the field of cancer. It relies on the release of these cancer cell associated components into the blood fluid of the affected individual, and they are used for early diagnosis with as much accuracy as possible. Furthermore, the nature of the biopsy helps us in organizing longitudinal studies. Longitudinal studies, like I had mentioned, basically means a time, like over a period of time, you can monitor a patient, and that helps you in like basically having a graph of how a patient responds to a certain diagnosis. Then we talked about how liquid I think my internet is not supporting. I'll switch it off. It's a little weak. Yeah. I had also discussed how they heavily rely on the process of epithelial to mesenchymal transition within cancer cells. And these were some of the questions that were posed. So this was a very quick, I rushed through the summary of the last class, but because we have a couple of topics to cover today, that's why I rushed through it. But if there are any questions about it, you can let me know. Otherwise, you can obviously refer to these slides, and that should help you get clear of a lot of doubts. If any questions, please let me know, else I'll proceed with today's session. Sorry, I couldn't hear you. I think that might have been accidental. Okay, we'll move ahead. Don't worry, I gave you a lot of information, I know, but then it was all covered in the last session. So either you can refer to the video, which I'll upload it. It's already recorded. I'll upload that and also give you the slides. So you can refer to that. And if it's a completely new concept or the entire field of non-invasive diagnostics, start slow. You'll find a lot of information online. Start slow and you'll understand it. It's an up and coming area of research, so you might not necessarily get a lot of information about it in the books. But yeah, if you are eventually hoping to work in cancer biology, this is something you should be pretty much versed with in the long run. Or if you're appearing for interviews and all, so then this would certainly help you. Okay, now today we are going to majorly focus on exosomes. We'll understand how they're relevant to cancer diagnostics. We'll understand very briefly about how isolation is performed. I think somebody had asked it in the last class, so we'll go through that. And finally, we'll get an introduction towards the conventional cancer therapies as well. I'll go slow now, don't worry. Just please interrupt and please let me know if something is not clear. This shifted a little bit, but okay. So, understanding exosomes. Now, what are exosomes? Exosomes are a class of cell-derived extracellular vesicles of endosomal origin, and they are typically 30 to 150 nanometers in diameter. They are the smallest types of extracellular vehicles. So, extracellular vesicles. So, these are commonly just briefly known as EVs. Extracellular vesicles are known as EVs, and they are a subset of all the types of EVs that can be formed. So look at this diagram for reference. Basically, yeah, I hope you're all familiar with the concept by which endosomes are formed. So basically, you have the cell membrane, which can sometimes pinch off. Sometimes, as in this is part of a process. It just doesn't happen randomly. It's part of a process whereby something from outside of the cell goes towards the inside by the process known as endocytosis, which means literally, if you just translate the terms, endo means inside, cytosis is it is going inside of the cell. It's taking something up. So during this process, it sort of pinches off like this. And as a result, whatever component was outside is now engulfed into a small vesicle. On the gestures and all. Just give me a second. Let me check. Yeah, recording is on. Just a second. Okay. Yeah. So it basically pinches off and it forms these smaller vesicles, which are known as endosomes. And within the endosome, lots of different types of components can be present. These are just representative. Next, what can happen is that these can become bigger in size, and sometimes endosomes they can form phagosomes, they can fuse with lysosomes, they can form phagolysosomes. All of those are that's an entire different domain of like basically vesicular trafficking and this endocytosis process and all. I'm not going into details of that. Basically, what you just need to know is that from a large multivesicular body, which is a late endosome. MVB is for multivesicular body. From that, these smaller vesicles can sometimes be formed, and these vesicles are called as exosomes. They are the smallest type of extracellular vesicle. Extracellular means something which is going to be secreted out of the cell. And these are typically around 30 to 150 nanometers in diameter. So you can imagine how tiny they are, and they are just small vesicles. What do they exactly compose of? They have a lipid bilayer. They are released into the extracellular environment, and they contain a very complex cargo of contents which are derivative of the original cell from which they have originated. And that includes the cargo includes proteins, lipids, mRNA, microRNA, and DNA. Exosomes are majorly defined by how they are formed through the fusion and exocytosis of multivesicular bodies into the extracellular space. That is what this diagram is showing you. So first fusion has happened, and as a result of that, this huge, larger vesicle has been formed, which is a multivesicular body. And from there, there has been release or exocytosis of these smaller vesicular bodies into the extracellular space. That is what is known as an exosome. Now, please keep in mind that exosome biology by itself is huge. It's not possible to cover everything about it right now. If you are unfamiliar with exosomes at all, a little bit of groundwork would be good for you to do on your end. I'm introducing the concept, but keep in mind I'm introducing it in the context of cancer diagnosis. By itself, exosome biology is very huge. So if it is something you are interested in, you would probably have to read a little bit. It won't be possible to cover so much in just this 2 hour session. But anyway, for now, let's understand some basic principles about exosomes. First of all, they are generated by budding from both the plasma membrane and from the endosomal membrane. Second, exosomes can transfer signals and macromolecules to target cells. So this is a very, very important context. They are not just, um, like passive bodies, not just passive vesicles which are pinched off and they're just carrying something. No, they're actually capable of relaying information from one cell to another, obviously by the process of fusion, or in some case, if exosomes carry something on their surface, a specific type of receptor on their surface, then they would be able to interact again with another type of cell which has the ligand which is specific to that receptor, and therefore there can be cell communication which can be mediated by these exosomes, and they can transfer some information, transfer some signal, and also upon fusion they can transfer molecules from one cell to another, some sort of signaling molecule. So it's an important job. Thirdly, exosome biogenesis under normal circumstances is a mechanism of protein quality control. So, if you have read a little bit about quality control measures, you would know that um, any cellular component which is synthesized is always kept in check. The synthesis process is kept in check by multiple mechanisms. We have discussed this I think in the first or second session, a little bit about it, that when we talked about positive feedback and negative feedback loops, which operate to keep on check on what signaling pathways should be switched on, and how they should actually be controlled. The switching on and off should be controlled. Not just that, they were just basic signaling pathways which are necessary for cell survival. But the production of molecules is also under very strict control. You might have read a little bit about it, that, think about the most basic processes about DNA replication, RNA production, which is transcription or translation, or the trafficking of translated proteins, protein folding. All of these are under lots of checks. If you've studied protein folding, you would know a little bit about quality control already. So, because folding is a very energy intensive process, so if a protein has any sort of mistakes in it, then the cell has multiple checkpoints to make sure that they're not unnecessarily spending a lot of money on trying to fold the entire protein, but instead it starts folding smaller domains and then it folds the entire protein as a whole. So again, this is a whole other domain that I'm going into. Coming back here, exosome biogenesis is this production of exosomes, basically is a measure by which proteins production is kept under control or is regulated, and this is a mechanism for doing that. Um, Finally, exosome biology, it plays very critical roles in human health and disease, which means that for normal cell function, for physiological function, exosomes are very important, and also in context of disease. So this is what we are more interested in, and that is what we'll be exploring a little further. Again, any doubts, any queries, you can please let me know at any point of time. Any queries till now? Then just unmute and let me know, and then we'll move ahead.
No, ma'am.
Okay. Okay, I'll move ahead for now. But yeah, if anybody has any doubts, just let me know. So, continuing with this theme of exosome biogenesis. So, this is actually showing you some contents of the exosome. Again, just focus on this diagram first. You can see the endosome, multivesicular body, and you can see that from the nucleus, some fragmented DNA is also a component or one of the cargo that is loaded into the exosome, and then it gets secreted. So, look at the legend first. Exosomes originated from multivesicular bodies containing many kinds of proteins, such as membrane transporters, Hitchcock proteins, and so on. In addition, also it contains a lot of non-coding RNA, like microRNA, long non-coding RNA, and circular RNA. These are very, very important in the role or in the development of tumor. So this is just showing you the full forms of the abbreviations. MBB is for multivesicular bodies. CDEs are for cancer-associated fibroblast-derived exosomes. MSCs are mesenchymal stromal cells, etc. So you can refer to that. But what this effectively is to show you is that what you just see as a simple circle actually has a composition like this. So what exactly do we need to note here? You can see a lot of information given. No need to, panic about it. Just look at details which are necessary for you to understand. First, what is the dimension? Like we mentioned, somewhere around 30 to 150 or 40 to 160 nanometers. This is the average dimension. The size will vary depending upon what sort of cargo it is carrying, right? Secondly, what do you see? The composition of the membrane is actually a lipid bilayer, which because it is derived, as you can imagine, from initially from the plasma membrane or from the endosome, all of which are bilayer in structure. And obviously, you can like logically also it makes sense. If it was just a monolayer of lipids, it would form a micelle. So, oops, just a minute. Yeah, so micelles, by their inherent nature, would not be able to host any cargo within them. They would just form a complete sort of ball of lipids or a monolayer of lipids by themselves. So, therefore, the lipid bilayer also allows for the presence of certain transmembrane proteins, integrins, or some sort of GDPase channels also to be present. So that allows for the interaction to also be feasible. If you remember, like I just mentioned, some of the receptors or ligand receptor interactions can be facilitated if it is present on the cell surface. So apart from these, you can see these transmembrane cross-linked proteins which are present. Some examples of this are given. Integrins I already mentioned. Then you have certain annexins or RAP GTPase to be present, which is helpful for trafficking across the membrane. You have intracellular components like HSPs, which are hitch of proteins, non-coding RNA, like I already mentioned. You can see all of these different examples given. MicroRNAs, all of these are microRNAs which are present. This is the way in which a microRNA nomenclature is actually done. So MIR is for microRNA versus LNC stands for long non-coded and CIRC is for circular RNA. So all of these are present, and you can see that microRNA can be derived from different types of sources also, like from fibroblasts, cancer-associated fibroblasts, or mesenchymal stem cells, immune cells, etc. So these, as you can imagine, this is not just a it's not a passive body. It is carrying so much information and so much important information, and depending upon where this information is eventually passed on to, the type of response will vary and the effect of the communication can therefore be understood. See, give me a minute. Yeah. So this is overall the composition, like I mentioned. And apart from that, there are also cytoskeletal proteins which are present. So they are like practically they are more relevant depending upon what type of phenomenon it is. But in our context, these microRNAs, the DNA which is carried, and some of these surface receptor proteins, they are more important. So we'll try to keep the focus a little bit more towards it. So yeah, this is what the exosome is exactly made up of. Now, now what is it? What is the role or what is the context in which we talk about when we mention it for cancer diagnosis? So basically, secretion of exosomes has been reported to be a lot more increased in cancer patients. Hence, exosomal markers are very potent and very actively researched targets for cancer detection. So again, going back to what we saw here, this is what I explained in context of normal physiology. Like in a normal cell, also exosomes are produced. In our context, when we talk about cancer, the rate of production or the overall exosome abundance in blood is slightly, like when I say slightly, I mean in practical application, it is slightly more increased. But if you just make a normal versus cancer patient comparison, then it is a lot higher in cancer patients. And also because it is derived from the tumor, it is giving you information about the tumor itself. So here you can see the sort of a flow chart, a cyclic flow chart of what is done. So from the patient, you get your clinical sample, you enrich for exosomes. Enrich basically means that from the sample that you have received, you're going to sort of think of it like filtering out all the unnecessary things and just increasing the yield of your object of interest. In this case, that object is the exosome. So you try to enrich or enhance its abundance. Then from the exosome, you extract all the proteins and nucleic acids which are associated with it, and then you study that, and then you figure out a way if you can utilize it for cancer diagnosis. And if possible, you also try to explore its potential in drug delivery, and then if it has that potential, then it can be used to send some sort of drug into the patient because it is derived from their own exosomes, and that will help in cancer therapy as well. So exosomes are very promising, very actively researched areas, and they have roles in both diagnosis and prognosis. And it's, I think, one of the questions I've put as the like one of the thinker questions I've put for today's class as well. So look at that and see if it is interesting to you. Here, I've cited some few common examples of the way in which you can actually figure out, or in the way in which practically it has been studied, that there is some sort of coherence between plasma exosomes and the tumor burden, which is present in an individual. So, first example is for oral cancer patients, whereby in very advanced stage, in stage 4 patients, they showed that levels of CD63 positive and caveolin positive exosomes were upregulated in patients, and the presence of these markers was found to be associated with poor prognosis. Now, this sentence, while it looks something very simple, it actually has a lot of research and a lot of evidence has gone into drawing this conclusion. I'll elaborate on this. Yes, Ankit, you were saying?
Ma'am, what does CD63, like, what does positive mean in
Okay, yeah, yes, I was just about to explain that. Okay, let's go back to the previous slide. That should help you. So if you see here, right here where I have given these examples, you can see here CD63 and here you can see caveolin 1. So basically what would have been done, let me just give you an example of this study. So for these stage 4 oral cancer patients, say you have 100 patients from whom the blood was taken, and among those, in 40% of the patients, the exosomes that they found from these patients, they found that those patients um, whose sample derived exosome carried this CD63 marker on the surface and that carried the caveolin 1 protein inside the exosome, these patients had poorer prognosis. So basically, what is done is after you have isolated the exosome, it has to be characterized. So this is what I showed here. Like from the clinical sample, after you have enriched the exosome, you figure out what exactly is present on the exosome. So there are different ways of doing it. You will have to separate the intracellular component of the exosome, like whatever is present inside, that you study separately. You check what proteins are present in that. You take the lipid fraction, which is the outer boundary of the exosome separately, which is this, and you study that. You check what proteins are present, you check what DNA is present, you check all of those different microRNAs, etc. So everything that you can find within the exosome, that becomes characteristic for that patient. So patient 1 has XYZ markers, patient 2 has XY, but some other marker, say M marker. So depending on this, you can classify patients. So every patient that has one particular marker in common can actually be separated into a group. So a study like this has taken CD63 as one of the markers and that caveolin 1 as another marker. So when both of these are present, then present and present in very high abundance, upregulated means it is very high expression of that is present in the exosomes of these patients, then they are having very poor prognosis. Poor prognosis means the time at which the disease is actually diagnosed in these individuals versus the response after that diagnosis has been done, that is very poor, which means that either they do not survive or they do not survive for very long, or they do not respond very well to the treatment. So overall future of that patient from that point onwards is not good. It's not positive. So that is how the conclusion is basically drawn. So were you able to understand, or anything else you want clarified?
Yes, ma'am, I understood.
Okay. Yeah, and what is CD? CD basically is something you would have known if you've studied immunology at some point of time. Basically, for now, think of them as cell surface proteins. CD stands for cluster of differentiation. So, if you have studied about Treg cells and all, you would know that there are CD4 positive, CD8 positive T cells. So, basically, they are cell surface proteins based on which classification can be done. And each cluster, it basically represents the type of antibody that that particular protein can bind to. I don't want to complicate it too much. Just think of them as certain cell surface proteins. That's good enough. Just it's the nomenclature. So that is how this study was concluded. Then, in context of prostate cancer, they found that high levels of this exosomal CD81, another separate marker, and high levels of prostate-specific antigen were observed, and that actually distinguished prostate cancer patients from benign prostatic hyperplasia and healthy subjects. So here this is a separate thing to consider. In this case, what you saw was that two different markers, both of them present on exosomes, were used to draw a conclusion about the response of patients. In the second example, two separate types of samples have been taken. So for that case, exosome CD81, which means this particular marker is present only on exosome, and PSA, which is prostate specific antigen, is usually measured from the blood. So a blood report and the exosome derived report, both of them were taken for patient categorization, and based on that, they could figure out very clear differences between a prostate cancer patient, BPH patient, and a healthy patient. This why is this important? That is because this is a very disease specific example. You don't have to worry too much about it. But basically, what happens is that in certain diseases, for example, prostate cancer, the lining between a healthy subject, a pre-cancerous subject, and an actually cancer patient is sometimes a little blurred. It's very difficult sometimes to figure out whether a person just has an inflamed organ, which is hyperplasia. If you remember very early on in our classes, we had discussed hyperplasia, whether a person just has this benign disease, whether they actually have cancer. This distinction is sometimes very tough. So there are multiple ways which are being explored and some of which have been standardized also. But one way was to look at the exosomal marker CD81. So that along with high PSA was considered to be a sure shot marker of prostate cancer, and it could be clearly distinguished from a normal or a precancerous stage. So this was another disease specific example. The third one it was delivered by a certain research group led by Jacobson et al., where they developed an extracellular array and they assessed the level of 37 lung cancer related proteins in exosomes. The assay was reported to have 75% accuracy in distinguishing NSCLC patient, which is non-small cell lung carcinoma patient, which is a subtype of lung cancer, from healthy individuals using only 10 microliters of plasma sample. So if you have worked in a lab, you would understand how little 10 microliter is. It's a very small sample amount. Um, Basically, just for context, if somebody is not familiar, 1 ml is equivalent to 1,000 microliter. So 10 microliter is obviously a very small volume of it. It's like 1/100th of it. So therefore, a very small sample size is good enough to give you this much confidence, 75% confidence. And therefore, that's something that's very promising and very much an area of interest for cancer diagnosis. So this is like another thing people have been trying to standardize, that you limit the amount of sample required and you try to make the diagnosis as accurate as possible. The effort is always towards that. And the reason for that is, if you remember in the last class, I'd already mentioned it, that from the patient blood, you might get, say, 2, 3 ml of blood, but from there, the actual plasma that you get, and from the plasma, you get the number of exosomes that you get is very small. Like it keeps on getting filtered. And so your effective sample that you use for study is extremely limited. And so, therefore, the lesser amount of sample required for a better diagnosis, the better. So these are just some examples. So just to give you some real life context about, how really are people, what are people doing with exosomes? But these are all reported studies. Of course, you can read more and more about it. These were taken from a review. I think I forgot to cite it. I'll put it when I upload the slide. But just if it's interesting to you, just go ahead and look at it. It's pretty cool. It would be interesting to read about. So yeah. So before I go into the isolation thing, any questions? Anybody has anything they want to clarify? Let me know, and then we'll move on to how exosomes are isolated. Just let me know if you are understanding it. These probably are unfamiliar concepts. So if I go by too fast, then just let me know, and I'll try to do my best to explain it better. Okay, if not, we'll move ahead. We'll look very briefly at the process for isolation. So, basically, what we have learned till now is that what are exosomes, why are they generated in the body, how are they relevant to cancer, and what information can they provide? This is what we have looked at. Now, the question is, you have patient blood, what do you do? How do you get exosomes? So I'll give you a brief overview. Don't worry, I'm not going into like details of the protocol or anything. But yeah, just to familiarize you with what needs to be done in the lab, in a pathological lab, or in like in a basically molecular biology lab. So exosomes can actually be extracted from very different sources. For example, from the cell culture medium. Cell culture medium basically means when you are culturing um, cancer cell lines in vitro. And okay, this is some. I'll just interrupt this for a second. There's something I was wondering, even though it's not a part of the flyer. If time permits and like if we can complete everything as per schedule, then in one of the classes, I would like to introduce the different types of models which are used to study cancer biology. So like cancer cell lines, and of course, you probably are familiar with it, but a little bit about organoids, a little bit about spheroids, the differences between them, and also mouse models, the type of mouse models which are used for cancer study. So I would like to introduce that. So it hopefully will be interesting and would be helpful for you. So we'll see that after we complete whatever is already planned, we'll try to incorporate that as well. So coming back here, the cell culture medium, body fluids, and tumor tissues, all of these can be used to actually harvest exosomes. The methods to harvest include differential ultracentrifugation. You probably know what it is. Ultracentrifugation means centrifuging at very, very high speeds using some specific type of centrifuges. And density gradient fractionation, which means that it separates like different components of the cell or of the fluid which you're using. They separate based on their density because a gradient of densities is used. Polymeric precipitation, which means you sometimes use certain sort of polymers like agarose, cephalose, etc., to precipitate or to pull down certain types of components. You can use ultrafiltration, which means you use a sort of filter of very, very, very minuscule microscopic sizes. So it will only allow a certain size of things to be retained while the rest of it will pass through. Size exclusion chromatography, and immunoaffinity isolation. So this is something we will be looking at in the next slide because that is more promising, more relevant, and more commonly used nowadays. So this is what you see. This is a representation of the way in which exosome isolation can be traditionally done. So you have your sample, which is subject to differential centrifugation. Like you can see, 300 to 500 G for 5 to 10 minutes, then 10,000 G for 30 minutes, and that helps you in separating the cell debris and the rest of the supernatant. Then this debris can again be centrifuged at much higher speeds of 10,000 G. G here is RCF for 30 minutes. So this is a long and a high speed centrifugation from where you actually get a pellet of debris and large extracellular vesicles. They would be separated, and the rest, what remains, is your supernatant. Then that is filtered through 0.45 or 0.22 micron filter, which means that think of it like a filter paper. That filter paper has very, very small pores, and those pores are around 0.45, 0.22 to 0.45 microns in size. So only components which are smaller than that would be retained and the rest would pass through. So then this filtered fluid or filtered supernatant would then be subjected to way higher speeds, which is a part of ultracentrifugation, which is 100,000 G. So you can imagine how fast it is spinning. So you might have seen these sort of centrifuges if you've like been to a lab or worked there. So you have sized centrifuges. So with that, this high speed can be attained, and such long centrifugation has to be carried out. And as a consequence of that, what you get are small EVs, and then you can wash them and again centrifuge them, and finally, what you would get would be exosomes. So a long process. Yes, Ankit, go ahead.
Ma'am, what would be the use of PBS in here in ultracentrifugation?
Um, I think it would just be to wash everything out. I assume it is just only for washing. It's like PBS in general is to maintain the pH. It acts as a buffer. Naturally, it's phosphate buffered saline. So it is to wash and to maintain the pH so that EVs stay intact. You don't want them to rupture at any point before this. Um, yeah, I think that is all because you start off with blood or media or some other fluid, and at this point you have isolated vesicles. Now you want the vesicles to remain or to not remain, retain their conformation, retain that bilayer structure, and thereon when you want to actually lyse them under whatever conditions are required to extract the nucleic acid and to extract the proteins, only then that should happen. So even if you do normal cell culture, PBS washing is done just to maintain the tonicity of the cells and the pressure within the cell and to maintain that balance. So it's a common step which is usually done, and also it helps in removing debris. So your cells would stay behind anything which is not like despite however much sterility etc you maintain, not even sterility, but however cautious you are, sometimes you would have some unwanted components staying behind. So for example, large some broken parts of larger EVs might have stayed behind, some cell debris might have stayed behind. So all of that gets basically washed off. It is another subsequent step to get rid of anything which is um, not desirable. But yeah, I've not done this myself, so I'm not very expert in this. If you want, I can go back and look at the need for PBS. But honestly, this is a technique which used to be done more commonly in the initial stages. There are better and quicker ways of getting exosomes nowadays. But yeah, this is the overall idea that I have. But I can go back and look at it if you need. Okay. Now moving ahead, this was the technique that like I mentioned, was something. It is slightly, I wouldn't say primitive, but it's a slower process, as you can understand. What helps in better confirmation and more, how do I explain it? Like a more specific subset of exosomes can be isolated by the technique using immunoaffinity isolation. So, as the name suggests, the name is good enough for you to understand. Immunoaffinity means that there are antibodies involved, and the affinity to antibody, which means that how strongly these exosomes bind to whatever XYZ antibody you are using, that will decide what type of exosome is going to get isolated. So you are using antibodies to filter out. Say, for example, from one patient, you might find 50 types of exosomes that might, I'm just giving you a random number. So say 50 types of exosomes can be found. From there, you only want those exosomes which have, say, for example, like we discussed, the CD63 marker on their cell surface. So you can use an antibody specific to anti-CD3, CD63. So that will help you to segregate only those exosomes which actually carry that expression on their surface. Keep in mind, this isolation is only going to be relevant when you are using characterization of a surface protein. So you cannot use an anti-caveolin 1 antibody because that is present inside the cell, and that is only possible. It's only going to be able to access caveolin 1 if you rupture the exosome and it's able to release its contents outside. So surface-based characterization is possible by immunoaffinity-based isolation. Um, if you are familiar with immunotechniques, you would know that most immunotechniques are actually relevant only in this context. Like, for example, if you've learned about FACS, which is fluorescence assisted cell sorting, you would be familiar with the fact that based on what type of protein is expressed on the cell surface, separation can actually be done using the necessary markers. So that's a separate context. Coming back here, so antibodies conjugated with beads can either select the desired exosomes, which is the example I was giving you, which is immunoenrichment. So that means, say, again, take that CD63 molecule. If you have anti-CD63 antibodies, that will only allow you to select those which carry that marker on their surface. That is enriching for CD63 positive exosomes. The other context in which it can be used is that you filter out those exosomes which carry anti-CD63 and you do not take them, and they are considered to be unwanted exosomes, and then that is immunodepletion. In both contexts, isolation can be performed. Now, this selection process makes it possible to clarify these unique exosome population, but of course, it leads to lower yield. So again, if you have 50 exosomes, like average of 50 exosomes per individual, again, random number, don't take these like very strongly. From there, if only 10 exosomes carry CD63 marker, then you are cutting down the yield of exosomes by a lot. Like from 50, you're getting 6, around 10% is what you're finally getting. So that is going to give you a very low yield, but it is going to give you a lot more specificity and a lot more. Um, it depends upon your project. If your idea, if your hypothesis is dependent upon working on one type of exosomes, it makes sense to go ahead and make this categorization. So it's an available technique, and it helps improve the accuracy and the science of your study overall. Apart from that, what I would also like to to understand is that how can you actually see the result of this isolation? So most commonly, Western blotting is used to detect exosomal protein. So here till now you've gotten the exosome, then you can lyse it and you can study it. So Western blotting is very commonly done to check what proteins were present inside the exosomes. I assume most of you are familiar with Western blotting or immunoblotting. Basically, it's a very commonly used molecular biology technique to detect specific proteins in your sample of interest. Apart from that, ELISA is also used, which is enzyme-linked immunosorbent assay for the detection of exosomal proteins. If in case anybody is not familiar, I will like when I upload the video for the last class and today's class as well, the recorded lecture. From there, you can go ahead and check. I have a previous class taken on immunotechniques as well, sometime last year, end of last year. So you can go ahead and check that out as well, or you can just refer to any available online material. But these are very basic immunotechniques. So if immunology or cancer immunobiology is of interest to you, just go ahead and brush up these topics. They would be very relevant if you are planning to work here in the long run. Now, traditional ways of detecting the exosomal nucleic acid that involves QRT-PCR, which is quantitative real-time PCR, microarray, and RNA or DNA sequencing. So people now go for RNA DNA sequencing over microarray, obviously because it has become a lot more cheap. It gives you more, it gives you high throughput data, which is going to give you a more comprehensive idea of what happens in this condition, and it is more relevant in today's time. It's more acceptable in today's time. So both things you are now familiar with. So for from the isolated or harvested exosome, you can detect the protein, you can detect the nucleic acid. So you have a lot of information. However, exosomal nucleic acids are often of low abundance. Obviously, like you understand, the number of exosomes themselves are so low, and from that exosome, you are isolating nucleic acid. So it's every step of filtering is going to reduce the yield or going to affect your the final concentration that you get in the long run. So it affects the sensitivity and the accuracy of detection. But to address these problems, I had already mentioned this in the last class. Droplet digital PCR is formed, which is a much more recent technique. It's not very new in today's time, but basically it helps in partitioning of PCR mixtures into these little droplets, basically, and within each droplet, PCR is carried out, and that helps you in measuring the reaction. So these are all very cool, cutting edge techniques. So if it is interesting, definitely read up on it, and it's something which is very relevant whenever you have to work with a sample which is um, very sparse, very low abundance. So in that case, this is a very, very sensitive technique and it works very beautifully. It can detect extremely low amounts of DNA and of specific mutations within that isolated DNA. So that's very, very cool. A little more detail about the isolation process. So if you see, this is just a stepwise representation. You can refer to this review. I've cited it below. It's a good review to get started if it is something you want to read about. So you have your conditioned medium. This is your cell culture flask. From there, a conditioned medium basically means the media into which exosomes have been secreted. So obviously, this would only be possible for adherent cells. So your cancer cells are present, cancer cells or whatever cells you are studying. Let's take cancer cells, for example. They are adhered to the bottom of the flask, and in the media, which is present above, they have secreted their exosomes because, as you remember, they are extracellular vesicles, which have been released as a part of exocytosis. So that entire media is collected and then that has to be centrifuged and you get the cell pellet first, you remove the dead cells. You can see multiple centrifugations which have occurred. Then you subject it to that filtration, which you saw from point 2 to 0.45. Then this is a separate step. You can use concentrators to um, basically concentrators are sort of, think of them like sort of filters. If you have performed mini prep, midi prep, etc., you know that there are these sort of columns, right? These columns they bind DNA, and then you can remove or elute that DNA in a much smaller volume. So from say a 1 liter culture, you can get DNA in 100 microns. microliters of sample. So that's similar concept. It's not the same. I'm just giving you the sort of extended reference. But what you try to do is you try to concentrate the DNA and concentrate the exosomes, and then you collect it from there and then resuspend it in PBS, which acts as a buffer in this case. So this is enrichment of exosomes from your culture medium. Next, this can be immuno-captured beads assay or that immunoaffinity isolation that I mentioned previously. So you use, so this is an additional step. You don't have to worry too much about it. If I'm giving you too much information and it's scary, don't worry too much. It's just schematic representation of what I have already explained. So basically, antibodies are biotinylated. Um, okay, so let's understand this. This is something which will help you in the long run. Basically, what you are doing in this step is you are prepping your beads. So what are these beads necessary for? Let me go to this third step first, and that will help you understand it. Uh, no, okay, let's come back here only. These little circles that you can see in yellow, they were part of this entire population, this multicolored population of exosomes. This is what you expect to be present in the solution you have isolated. So from the first sample that you took, whether it is condition medium, whether it is patient blood or some other sample, from there, all you enrich for are exosomes. Bring the analogy to the same as PCR or think of Western blotting. All of them pretty much have the same idea. For Western blotting, what you do, you first isolate the total protein of the entire cell. From the total protein, you only detect your protein of interest using an antibody, right? Exactly the same concept. From all of the exosomes that you have isolated, you try to look out or you try to filter out your exosomes of interest. So to do that, what needs to be done, you need to have a strong support structure or like a strong scaffold. How is that scaffold built? That is built using these bigger beads. So say, for example, if you just had floating antibodies in solution, that would actually be difficult to separate out because antibodies are not very heavy. How would you actually separate it out? So for that, this sort of support structure is used, which is a streptavidin bead. So there's a 50 microgram streptavidin coated bead. So this bead is then this bead is actually bound to antibodies using biotin. So if you have studied some context about this, from like prior knowledge. So biotin and streptavidin have a very strong non-covalent interaction among them, and that interaction is extensively used in molecular biology. So biotin by default binds to streptavidin. So you sort of form an indirect connection between the three of them. What is your objective? Let's realign everything. You want to get antibodies which will bind to your exosome of interest, and that will be separated out. How do you do that? You mobilize or you sort of mm, provide a sort of a structure onto which your antibodies can bind, and then you pull that out. So for that, what do you need to do first? You need to bind your antibodies to biotin. This process is called biotinylation. This biotinylated antibody can now bind to streptavidin. So this is forming a much larger structure. So what will happen now, instead of one exosome binding here and one exosome binding here, so many exosomes can all be localized onto one single support. That support can then be separated out. Okay, that is the concept behind it. If anybody has any doubts, just let me know. I will explain this once again and I'll like go through the flow in the way it was intended. Just let me know. Did you understand it? Then only I'll move ahead.
Can you explain this once more?
Yeah, okay. I thought it would be confusing. Okay. So, give me a moment. I'll just switch on the video. Maybe that will help. Uh, okay. So basically, think of it in this way. You have different types of exosomes, like I already mentioned. You take a patient's blood, and from there you will get, let's stick to the numbers we had already discussed. You will have, say, 50 exosomes, 50 types of exosomes present there. You want only exosome X to be all the exosomes which carry protein X to be separated out. This is your objective. The question is, how will you do that? So you would have to use an antibody to separate it. You would use an anti-X antibody, which will bind to those exosomes and will separate them out. This is the logic behind doing this experiment. Now, how do you do it practically? Say, for example, if you had just used this patient's blood derived exosomes and you added antibody X to it, what would happen? The exosome and antibody would bind. Only that would happen. Now, how do you get that out of the solution? You have to separate them, right? You only have a mixture which has all of these proteins and all of these antibodies present. To separate that out, what you do is you create a support system. So think of this as the bead. You have a strong support. That support is the streptavidin bead, which is shown here in black. On this, onto this support, you want to bind your antibodies of interest. So what will you do? Think of it like a magnet. You are creating a magnet for antibody X so as to separate it out. Now, to create this magnet, there has to be sort of a linker because these antibodies will not directly bind onto the bead. You need some sort of a glue to attach them. That glue is this biotin shown here in red. So what you do, think of it like you have one, like say for example, this is your antibody. You attach biotin onto it, which is your glue, and then you stick it onto the streptavidin bead. Now what has happened, this entire bead is filled with all of these antibodies on the surface. So you have created this giant magnet, and that magnet will now be added to your exosome solution, and that magnet is going to attract all of the proteins or all of the exosomes which have protein X on their surface. So you are now going to get an entire population of only protein X. All the non X chromosomes or non X exosomes are not being will not be able to bind. Only those which have X on their surface will be able to bind. The rest of them will be separated out. Did it make sense now?
Yes, ma'am.
Yes, ma'am.
Okay,
Thank you.
understood. Okay. So this is what is happening. You have this magnet with all of your proteins of interest which are attached. These proteins of interest are coming from your exosome that we were actually looking at. So this I just explained in layman terms, but for your understanding, you should know the jargon, the scientific terms you should know. So basically, what you are doing an immunocapture beads assay. Immunocapture. What does it mean? You are using antibodies to capture something, right? What are you capturing? In this example, you're capturing exosomes with protein X. That is what you're capturing. How are you capturing it? Using anti-X antibodies. Okay. And what are you using in this process? You're using these giant beads, which are acting as the support system. Okay. Now, what are the sub steps? I'm just explaining the jargon one by one. Look at it in your own time. You'll understand everything. It's just new, so it feels like a lot. First, what are the jargon here? You have antibodies, anti-X, like I mentioned. You have biotin, which is your glue. What are you doing? You are adding the glue onto your antibody. The process is known as antibody biotinylation. Biotinylation means simply addition of biotin. That's all. Okay. Next, what you are doing, you are adding this onto your bead. What is this process known as? Functionalizing the bead. Because by itself, the bead cannot do anything. It's not. It's just a heavy protein which is present, and it's not going to do anything. It's just going to settle down. What you're doing is you're making it functional, and you're making it functional as per your need. For that, you are binding these glued antibodies onto the bead, and you're creating this strong structure. So this is a functionalized bead. How do you do that? You just allow them to interact for 60 minutes at room temperature. This becomes your, like I mentioned, the magnet, which is going to work in this case. This you add to your collected exosomes or your enriched exosomes, and you incubate it overnight. Matlab you allow them both to interact overnight with rotation at 4 degrees. So basically, in a solution, think of a giant water bottle you have. Within that water bottle, you have these beads, you have exosomes, and you just keep rotating that water bottle overnight. So they'll constantly keep coming in contact with each other. And wherever that interaction is specific, wherever these exosomes are able to bind, they will get localized. Now, if you have studied antigen antibody interactions, you know that these interactions are very strong and they do not detach very easily unless you're using some detergent to get rid of it. So, such interactions, such strong interactions will be captured overnight. So, what you have as a result is an immunocaptured subpopulation, which is of exosomes. You have a captured subpopulation with your protein of interest. This is what we have done till now. I gave you a lot of detail because if you study any immunotechnique in the future, that you'll just this will become very simple for you in the long term if you study immunoprecipitation, if you study biotin streptavidin interaction. So there's a lot to learn just from one schema. So I went into details about it. I hope it helps you. Now, what you do? Now you have your magnet, which is bound to the exosome. What you want now? You have to go back to your original objective. You want only the exosome. You don't want the beads. You want to separate the exosome from that bead which is carrying it. So for that, magnetic separation is performed. Uh, in this case, magnetic separation has been given because magnetic beads are commonly used, but depending upon what type of bead is used. So again, just to give you a little bit context, there are these sort of beads which are available. So you can just use a magnet stand and you have your tube which has these magnetic beads. So if you bring it close to the stand, all of the beads separate out and you can just remove the solution. So this is used, it's expensive, but it's used. They're called Dyna beads. Alternatively, if protein beads are used, you can just give it a spin, you can centrifuge it, and the heavy beads, they settle down. Different ways of extracting it. And from there, you elute using acidic glycine, pH 2.7. So that helps in removing these weaker interactions. And from there, you centrifuge, you take the supernatant, you collect it. Multiple, these are all protocol specific steps. You don't need to stress too much about it. But finally, what you do is you elute only the exosome. So the bead stays behind, the glued antibody stays behind. From top of the antibody, only you remove the exosome of interest. So don't stress too much about this. These are conventional manual techniques which are given. Nowadays, you have lots of kits available. They are wonderful, give very high efficiency, very low contamination yield, and they're extensively used. But the basic idea is this. So just look at the legend once for your understanding. So in step one, we had looked at the enrichment of exosomes from cell culture using differential centrifugation. Next, you are doing immunocapture using those beads for selective isolation of exosomes. Third, you are eluting them. Elution, these are again important terms. Washing and elution, two very different things. Washing means you are removing any contamination, any undesirable things from a mixture. So, for example, if some non-specific wrong interaction has happened, then you want to get rid of it. That gets removed during washing. Versus elution means the molecule that you want, your protein of interest or your um, say for example, if you're isolating DNA or you're isolating RNA, in that process you only want that molecule of interest to be removed. So that step in that process is called elution, or you are eluting your protein of interest. Don't mix up the two, they mean completely different things. So elution or extraction or removal of the immunocaptured exosomes by magnetic beads. So that is what has been indicated here. So just go over it very carefully. I am pretty sure you will understand it. It has been quite well explained, but this is the overall protocol for isolation. So if it was clear till now, just let me know, and then we'll move on to conventional routes for cancer therapy. Any doubts, any part that was unclear, please unmute. Please don't hesitate. Any doubt, please ask me.
No, ma'am.
Okay. Uh, Other people, anybody, some of you are a little quiet. It's okay. If it's your first class or something, it's okay. But if any doubt, please let me know. I'll try to address to the best of my capacity. Okay, so this completes what we had to learn about invasive, non-invasive diagnostics, which are the current, the hotcake topics in the field of cancer diagnostics. And if anybody is interested, you definitely need to read about these. It will give you an edge above others. Now we'll take a step back. This was one of the topics left behind from one of the previous classes when we had briefly discussed about cancer therapy. So we are going to go over the conventional routes very quickly. And then in the next class, we'll talk about advanced, or not even advanced, the current strategies for cancer therapy and how improvement has happened in cancer therapeutics in the last several years. So whenever you hear about a person, like we all know somebody who has suffered from cancer. So most commonly, the strategies that you hear of are the most conventional routes. They are what have been tried and tested and have been approved over time. However, it comes with that huge, you know, sort of a warning or a side effect that comes along with it is that it is not very specific, and there are going to be lots of off-target effects or non-specific effects, and that is something which people just deal with. There's nothing to do about it because that's how the procedure works. So what are the three conventional routes? The first one obviously is surgical therapy. What does it mean? You perform surgery and cancer cells are removed. Second, radiotherapy, which is radiation, whereby the diseased part is irradiated, whereby it is subjected to very strong radiation or very strong ionizing radiation or basically beams, which help in killing the rapidly dividing cells. Third is chemotherapy, whereby you take a chemical, which is a drug or a medicine, and that medication is used for therapy. So these are the three major strategies. I'm not going into details about radiation because it will take the whole discussion in a different direction. We'll briefly discuss surgical therapy and then move on towards chemotherapy, the most common treatment we hear people getting, and the most common treatment which we know has so many side effects. The first one anybody can think of is that when somebody is undergoing chemo, they lose all their hair, right? So we are more familiar with it. So we'll go into that discussion soon. Let's start off with surgical therapy. So, what types of surgical therapy can a cancer patient undergo? So, it's not just any surgery. There are different subcategories within it. So, I want you to like get a little familiar with them. The first one is obviously curative surgery. What does it do? You remove the cancerous tumor or the cancer growth from the body. When is it performed? This is only recommended and only performed when the cancerous tumor is localized to one specific area of the body, which means that cancer is still under control. It has not progressed indefinitely, and it is not in a very advanced stage. This type of treatment is often considered as a primary treatment. However, other types of cancer treatments like radiation may be used before or after the surgery. So it is often given as a combination. Surgery plus radiotherapy is often recommended to patients if they have a localized sort of cancer. Again, keep in mind that radiation would only be feasible in that situation. So if somebody has a disease that has progressed very far and like they have tumors throughout their body, through their bones, in brain mets are present, lung, liver mets are present, then in that case, how do you subject that person to radiation? You would have to literally, you know, uh, treat the entire person's body with radiation, and that is going to make things 100 times worse a lot faster because these radiations are very harmful for the body, and they are only relevant or only used when you are only subjecting one area of the body because you know that is very rich in cancer cells, and therefore the cost benefit ratio in that case is something that surgeons or oncologists they choose to go for because that will give the patient a better shot at living. But yeah, it's not the best route to go for in all situations. Second, this we talked about curative surgery. The second subtype is preventive surgery. What is it done? When is it done? So, preventive surgery is used to remove any tissue that at this moment does not contain cancerous cell, but that can eventually develop into a malignant tumor. For example, in a very common situation where it happens are polyps in the colon. They may be considered as precancerous tissue, and preventive surgery can often be performed to remove them because these can eventually become cancerous. Another context in which this is quite often done is for ovaries also, or if somebody has some precancerous sort of lesions in the breast. In that case, sometimes surgery can be recommended. It depends very, very much on the cancer type, and sometimes it is recommended to remove it than to go for watchful waiting and then keep on checking whether or not it can become cancer. It's very context specific, but preventive surgery is done in a lot of cases as well. The third can be diagnostic surgery. That is because you remove, it's similar to biopsy as well. So you help, it is required because all the other tests that we have already discussed, like for example, lab tests or these non-invasive methods, blood tests, all of these, they are not conclusively um, giving us information whether or not a person has cancer. So in that case, diagnostic surgery may be performed whereby a sample tissue can be removed and then evaluated, and then based on what they are actually seeing in that surgery, they would be able to confirm whether or not that person has cancer, because that is the final stage, right? You're literally taking out tissue from the person to validate whether or not they have cancer. So that is a diagnostic form of surgery. Next, this is a lot of text I know, but then there's not really much image to show in this context. So just we'll go over it quickly. You have staging surgery, which is pretty similar to diagnostic surgery as well. So in that case, the objective was to just to confirm whether a person has cancer or not. In some cases, it might be slightly less invasive, whereby laparoscopy can be performed. Through laparoscopy, basically uses some probes through a small incision, these probes are inserted into the person's body depending on where they are suffering from cancer, instead of cutting them open and just taking the entire tissue out. So in that case, you can use a small mass or a small chunk of the tissue, and then use that to figure out what stage the disease actually is in, and what should be the approach for treatment. Next can be debulking surgery, whereby you remove a portion of a cancerous tumor and not all of it. Why so? Because it might be that if you remove the entire tumor, that can actually cause damage to the organ or to the individual's entire body. And what is in that case relevant is that, say, for example, if somebody has say pancreatic cancer, right? So, and that cancer has spread to a large percentage of the organs, say beyond 50% it has spread. Then in that case, it's not possible to remove the entire pancreas. And you can see that it has sort of spread all around the organ. You cannot remove the pancreas. I mean, that would not be the primary mode of treatment, because in that case, the person has to undergo a lot of lifestyle changes and a lot of other problems. So in that case, it might make more sense to remove some part of the tumor and then irradiate the surrounding area, so that you don't cells from that localized area do not spread and cause a much worse cancer. So in that case, you're just reducing the tumor load, but you're not completely curing that person. So these are all, these are oncosurgeons who are much better experts at this. Radiation specialists and oncologists, they recommend based on how severe the disease is. um, this is palliative surgery. So I had introduced palliative care, I think, in the last class, which is like, it's a symptomatic treatment, basically symptomatic, or just a way of making things comfortable for patients, because it's not possible to cure cancer. So it relieves discomfort, or it can correct other sort of problems that the cancer treatment may have created. So it's just sort of a management sort of care instead of a curative care. Then supportive surgery can also be performed. It helps other cancer treatments work effectively, like insertion of a catheter. It's why you might not exactly think of it as a surgery, but then any sort of procedure um, which creates an incision or a cut in a patient's body would effectively be considered as supportive surgery. Finally, restorative surgery sometimes is used as a follow-up to curative or other surgery, so as to restore a person's appearance. Like, for example, for women with breast cancer, sometimes they have to undergo mastectomy, right? We are familiar with this. You might have seen in news about a lot of actresses who had to go through that because they carry a certain mutation in the BRCA1, BRCA2 gene. So they choose to undergo mastectomy just to ensure that they do not eventually develop cancer, and do not develop it at a stage where it is so, do not get diagnosed at a stage where it is so serious that it is untreatable or it is life threatening in that case. So then in such cases, they undergo a breast reconstruction surgery to restore their physical appearance. Sometimes for oral cancer, it is required because a lot of people might lose a huge chunk of their the area surrounding the masseter muscle, sometimes part of their tongue. So these type of, like, in a lot of cases, tongue, it has to actually be removed. So in those cases, these restorative surgeries are often done. So these are just examples. They are all part of the umbrella term which you commonly know of as cancer therapy. So I don't think there's a lot to ponder about that. So you can just read about it in more details if it is interesting to you. And we'll move on to the last topic for today, which is chemotherapy. So, uh, what is chemotherapy? The basic definition, obviously, as you know, it involves chemical molecules, which are drugs, basically like they're actual drugs or actual entities which are responsible for treatment. It's not radiation, it's not a procedure, it is a chemical drug which is responsible for this. What is the aim of chemotherapy? If we talk about it a little broadly, then chemotherapy aims to inhibit cell proliferation and tumor multiplication. Effectively, it helps in avoiding invasion and metastasis. So the broad goal is to, I think I discussed this sometime in one of the previous classes as well, that basically what it targets is rapid, random, incorrect cell division. That is its main job. So what does it help in, or how does it actually function? Is that it is going to rely on the property of cancer cells to divide way faster than other cells of the body, and therefore it ensures it. I mean, it technically is supposed to ensure that only cancer cells die and normal cells stay fine. But reality is quite different, and that is why chemotherapy has all of these different problems which are associated with it. So that is what is expected. However, this property of chemotherapy results in very toxic effects due to its effect on the normal cells. Now, inhibition of tumor growth can occur at several levels within the cell and its environment. Like, for example, traditional chemotherapeutic agents, they rely like they affect either macromolecular synthesis, or they rely on disrupting the function of neoplastic cells by interfering with DNA, RNA, or protein synthesis. So basically, they target all of these crucial processes, and that eventually is expected to lead to cell death. And cell death, in a lot of cases, for normal cells might also get delayed in that case. And the cytotoxicity means cell death, cell something which is toxic for the cell, that is most significant during the S phase, as it is the DNA synthesis phase of the cell cycle. Apart from that, vinca alkaloids and taxanes act in the M phase and block mitotic spindle formation. Okay, a lot of information given. We'll understand it better using the schema. But the crux of the story is that chemotherapy relies on inhibiting cell proliferation, inhibiting the ability of tumor cells to increase in number, to multiply and increase in number. And the main problem associated with this condition is that it effectively leads to a condition whereby normal cells are also getting compromised along with your target cells. So let's look at the schematic to understand this a little more simply. So, basic principle of chemotherapy, as you can see, you have cancerous cells, and somewhere around it, you also have healthy cells. And drugs are going to kill cells which are randomly dividing. However, as you can see, induced cell death also affects the surrounding normal cells, and that is the problem that we have to address. Uh, what does it target? It target the targeting using chemotherapy induces cellular stress, it suppresses cellular defense, it initiates apoptosis, and obviously kills cancer cells. What is the objective? You want complete remission. Remission means that you want the person to completely go back to normal and not to have cancer anymore. You want to prolong their life, you want to improve the quality of life, you want to avoid recurrence, which means relapse of the disease. You don't want that to happen. And finally, of course, you want to provide comfort to the patient so that they have a relatively normal lifestyle again. So that is the goal of chemotherapy overall. There are different classes of chemotherapeutic drugs. First, let us look at okay, let us look at it in the context of the cell cycle. So obviously, I assume everybody is familiar with it, but you have these four major phases to look at. You have G1, S, G2, and the M phase. So during the G1 phase, what happens? It's sort of a preparatory phase. So there's a doubling of the cellular content. So there's duplication of the genome, duplication of all of the cell organelles, of the lipid bilayer, all of this, everything is undergoing duplication. So during this phase, drugs like anti-neoplastic enzymes, hormonal drugs, nitrosoureas, all of this can be used for inhibition. Next, from the G1 phase to the S phase, there can be use of topoisomerase inhibitors. Now, I assume everybody knows where topoisomerases work. Basically, they are very crucial to the process of DNA replication. They open up DNA. Um, like the double-stranded DNA is sort of opened up so that enzymes can access it and DNA replication can actually be carried out. So if you use topoisomerase inhibitors, you are stopping the opening up of DNA and therefore DNA replication comes to a stop. So cell cycle is abruptly ended here and cells can no longer divide. So that is another way of stopping cell division. Apart from that, you can use antifolates, antipurines, antipyrimidines, antimetabolites, all of this, which are all parts of the entire process of replication. If you target those proteins, you will inhibit the S phase of the cell cycle and cell division will be brought to a stop. Third, to target the G2 phase, you can use DNA check or DNA, you can target DNA check or DNA repair proteins. So alkylating agents, platinum agents, and tumor antibiotics can often use. However, they are considered to be non-specific drugs because they act in a more generalized manner, but they are quite promising if we look at it just in this context. Then finally, during the M phase, two major groups actually are used: the vinca alkaloids and the taxanes, which inhibit this process of mitosis or meiosis, basically the exact step of cell division, and they act in the M phase and they block the mitotic spindle formation. So I hope everybody remembers how spindle formation is extremely crucial for cell division. It helps in the assembly of the microtubules and then the separation of all of the material, but majorly the chromosomes, equally between two daughter cells. So that process is compromised in this case. So yeah, these are all of the different types of drugs. And here you have a little bit of insight into immunotherapeutic drugs. I kept it because it was part of the like same schematic, but it's not really relevant to us. So we'll keep it away for now. Immunotherapy is something we'll look at either in the next or the next to next class, depending on how the pace goes. But yeah, it is one of the newer interventions in cancer therapy, the more advanced techniques. So we'll come to that a little later. Here I have given you a little more detail about how these drugs work. So keep the legend, read the legend first. So you have the mechanism of action of some of these drugs, platinum and taxane-based drugs. So you have vincristine, paclitaxel, oxaliplatin, and cisplatin. Obviously, these two are platinum drugs. Paclitaxel is an example of a Taxol. Again, I would very, very strongly recommend if any of you are in that stage where you want to interview for, some PhD or some sort of a job in the field of cancer biology, please, please memorize all of these things. I was asked this question personally, how do these drugs work? So, and I was not able to answer one of them. That's why I really want to stress on you having the most basic ways of treating cancer very clear in your mind if this is the domain you want to work in the future, because this is what you're expected to know, most definitely know. Okay, so pay a little more attention to this. It's very simple, but you must know it. That's all I'm saying. So first of all, you have vincristine. What does it do? It prevents microtubule aggregation. So here you have microtubules, a representative example, and you can see that vincristine has been indicated in this context, whereby it in excuse me, sorry. So whereby vincristine helps in inhibiting microtubule aggregation, which means the formation of microtubules. On the other hand, you have paclitaxel, which works at the other end and it inhibits microtubule disaggregation or the disassembly of microtubules, both of which lead to cancer cell division arrest and cell death. What do they disturb? They disturb that dynamic instability which is present for microtubules. For this, you definitely have to know cytoskeleton, which again, I'm assuming you are familiar with. If not, these are basics you need to brush up on. These are very crucial for cancer biology or for molecular biology in general. So these affect the microtubules. Second set, oxaliplatin and cisplatin, they bind to nuclear DNA, which is deoxyribonucleic acid, just the full form for understanding sake. What does it do? It causes disruption of DNA replication and transcription and subsequent arrest of the cancer cell division. So this is what you see here, that this is the replication fork and you have DNA RNA polymerase present, and this process is disrupted. DNA replication and transcription is disrupted by these platinum drugs. What does it lead to? It leads to cell cycle arrest and eventually cell death. Um, yeah. The DNA adducts, they activate, these are DNA adducts that are formed eventually. They activate apoptotic pathways that induce cell death and tumors degradation in the long run. Third, this is the third set you see. All four anti-tumor agents alter the function of mitochondria. Which four? All these four that you just saw: vincristine, paclitaxel, oxaliplatin, and cisplatin. They alter mitochondrial function, followed by disruption of the respiratory chain, which is the mitochondrial respiratory chain you already definitely are familiar with. And there is increased production of reactive oxygen species. Also, it damages the oxaliplatin and cisplatin. They damage the sorry, cancer cell mitochondria by binding to the mtDNA and altering its replication and transcription as well. Now, mitochondria are very, very crucial for cancer cells. Why? Because they are rapidly dividing. What do you need for division? A lot of energy. Who produces energy? Mitochondria, right? So there's a huge dependence on um, extreme functioning and like hyperfunctional and abnormal and increased mitochondria within cancer cells. So you can see that all these four drugs, they are affecting the respiratory chain, which is leading to increased ROS production. And simultaneously, these drugs also inhibit replication and transcription of the mitochondrial genome, all of which finally affect its function and activate apoptosis. So these are basically multi-pronged strategies, as you can see, all of which are leading to eventually cancer cell death, but they are, merging down to the same result, but the primary effect is through multiple mediators. Also, I think this is something you definitely should keep in mind, because you might be, whenever you're asked about the mode of action of these drugs like paclitaxel or vincristine, you might just talk about microtubules, but you definitely have to keep in mind what is unique. This is unique to these drugs, but this is common to all four of them. So these subtle differences you should familiarize yourself with if it is relevant to your career in the long run. And finally, in in part D, all four agents, they cause the activation of immune cells, which effectively gives rise to tumor cell degradation. Like, for example, the macrophages, T cells, monocytes, all of them, they help in release of some pro-inflammatory cytokines, and they all trigger cell death. Eventually, all of them are just killing the cells in one way or another. They are just doing it through different pathways. So these were some examples of some of the drugs which work. And if we go in a little more detail about it, I think this is probably the last slide for today. Yeah. So finally, in the last slide, we are looking at a little more detail about the mechanisms of chemotherapy. So the first one is for cisplatin. So cisplatin is a platinum drug, like we just saw. It is transported into the cell through some copper transporters, and it is aquated. Aquated basically means comes in contact with the polar aqueous environment inside the cell. Aquated cisplatin, it induces DNA damage. Like you can see here, mitochondrial DNA damage and nuclear DNA damage. Both of these are affected. On the other hand, it also binds to cytoplasmic structures. Like what? Like reduced glutathione, which is shown as GSH, and metallothionein, which is shown as MT. Effectively, this leads to the generation of ROS, or reactive oxygen species, which trigger the mitochondrial outer membrane permeabilization, which is MOMP. Now, if you've studied apoptosis previously, you would know about the extrinsic and intrinsic pathways by which cell death is triggered. So, what it effectively does is that when there is rupture of the mitochondrial membrane, it releases the signaling molecule known as cytochrome C, which triggers apoptosis. and it is a mitochondria dependent pathway, and therefore the entry of this drug is finally leading to apoptosis in the long run. So this is just showing you the mechanism, the intracellular mechanism by which the drug actually works. Okay, I hope it is clear. It's quite straightforward and well represented in the schematic, so I guess you would be able to understand. Second, follow the legend again. 5-fluorouracil. What does it do? It can be transported into cells by uracil transporters due to its uracil-like analog structure. So you know that uracil is um, one of the bases which are incorporated into RNA, not in DNA. And this uracil-like molecule, which is known as 5-fluorouracil, can often be transported because it looks familiar. It is often transported into the cells. That is what you see here. Intracellular 5-FU or 5-fluorouracil gets converted into three active metabolites, eventually which lead to DNA RNA damage. What are they? The first one you see is fluorodeoxyuridine monophosphate or FDUMP. What does it do? It inhibits thymidylate synthase. What is this enzyme doing? It is responsible for synthesizing thymine in the cell. Why is thymine needed? Because more and more thymine would be required for DNA replication to actually be possible. So, and also because it is required for like basically when replication is occurring in a cell, all the DNA bases should be available within the cellular environment. So when thymidine synthase, shown here as TS, gets inhibited, there is an imbalance in the DNTP. Therefore, DNA replication will not occur properly, and eventually DNA damage will happen. The second one, FDUTP, which is fluorodeoxybane triphosphate, that is misincorporated into DNA because it looks very similar to thymine. 5-fluorouracil, after getting converted into FDUTP, which is the triphosphate, looks very similar to thymine, gets incorporated into DNA, but that is a wrong molecule that gets incorporated, and therefore that results in DNA damage and DNA replication comes to a halt. And 5-fluorouridine triphosphate, which is FUTP, it gets incorporated into RNA. So the same molecule works in three different ways, all of which affect DNA, RNA, basically nucleic acid uh, gets targeted in one way or the other. Its replication or its synthesis is affected, more and more damage is created, and all of that eventually leads to apoptosis. Now you might say that for cancer cells, we have seen that they have a lot of measures by which they are able to bypass DNA damage. Yes, normally they are able to do so. However, what you are doing externally is that you are increasing the load of DNA damage into the cell within the cell to such an extent that the cell is no longer able to survive. You are shutting down DNA replication, you are causing RNA damage, DNA damage, you are reducing available nucleotides. Effectively, the cell will eventually die. So this is the second mechanism. The third one is for docetaxel or paclitaxel, like we've already discussed. So it it stabilizes the microtubule, which is a huge problem considering the normal state of the microtubule is to remain in a sort of imbalance or an unstable state. So that stabilization is actually worse. And effectively what it does is it leads to apoptosis. So all the mechanisms are eventually merging to the same result. What is that doing? It is leading to cell death. So I hope it was clear to everybody. I'll discuss the key takeaways, and if there are any more questions, then you can go ahead and let me know. And after that, we will wrap up the class for today. Just give me a second. Yeah, I think somebody left. Yeah, no problem. So the key takeaways for today. First, exosomes are a subset of extracellular vesicles with immense potential in non-invasive cancer diagnosis. Already discussed this in the last class. Second, their isolation and detection remains a major challenge, but it is a very promising strategy for preliminary screening and diagnosis as well. Traditional routes have focused on killing the actively dividing cells or their complete removal by surgical measures. Chemotherapeutic strategies, while used extensively even today, they have severe major limitations associated with them, and there are a lot of off-target effects as well. The questions I would like you to look into a little bit. First of all, which hallmarks of cancer are explored in in conventional chemotherapeutic strategies. Think about it a little. It's not one hallmark, but a lot, multiple of them. Second, what are the limitations? We have already discussed it, but just go over it in a little more detail. We've just discussed the non-specific aspect of it. But yeah, what are other challenges that are associated with the same? And also, why are they still implemented in clinical practice if they have so many shortcomings associated? Third, apart from what we have discussed, which is the context of exosomes in diagnosing cancer, apart from that, what are other applications of exosomes in disease biology, and what are the associated limitations with it? So these are the key takeaways that I would like you to explore a little bit. Now, if there are any questions or any discussion that has happened throughout the class, please unmute, please go ahead and let me know.
No, ma'am, no doubts.
Okay, anybody else?
Ma'am, no doubts.
No doubts. Okay. The other two students. I think one of you, Abhi, I think has joined today. Um, yeah, it might seem a little unfamiliar to you if you're not from the field. But just go over the slides, or if you have enough time, you can go through the video lectures as well. Um, I think that should help you. But if there are any doubts from today, you can let me know. And the other student whose name I can't see, any questions, please let me know. Otherwise, we will wrap up for today. You're not audible. Yeah, go ahead.
Ma'am, can we treat cancerous cell into normal again? Because institute like Korean Institute of Sciences publish a research about that.
That is, I think it was a fairly recent research last year or last year, something like that. So people have actually been trying to do that to reprogram cancer cells. Um, See, first of all, it seems like a big claim. I will acknowledge the fact that they are experts in the field, and people will obviously try to, make it work. And I must say that nothing is impossible. So we don't know what is going to happen 20 years from now. It might be possible. People might, figure out a way to train cancer cells to become useful to us, useful to the body, re-inject them in some way or another. It's all possible. But theoretically, if we just think about it, a cancer cell will, by its very nature, should only learn to become worse for its host, to take over the host system for its own advantage. But yes, this is cutting edge research. And I think also one more thing that comes into question is the ethics associated with such research. So we don't know how far that can actually be implemented for another human being, can actually go into clinical trials. So they remain open-ended questions, and hopefully in the long run we might see something interesting. You never know, it might just happen that one person's cancer cell is brought into normal and then re-injected into that patient to elicit some sort of an immune reaction and clear out all the cancer cells. It's all, science is open ended, but based on what we know to be tried and tested, it seems like a far cry for now. That is all I can comment for now. Okay, any more questions? If not, then I'll end the class for today.
Thank you, ma'am.
Okay,
Thank you, ma'am.
okay. Then thank you. Have a good evening. Have a nice Holi tomorrow. We'll meet again on Monday, which is 17th, and we'll continue the discussion. We only have very few classes left, I think five classes, out of which four we'll use to wrap up like the discussion that I wanted to have from my end. And then the last class will be just a summary and all the questions that were left behind. So yeah, we'll wrap up this course pretty soon. So please keep joining. I was getting very low attendance for the last couple of classes, so try to attend the sessions. It makes it more interactive. But yeah, thank you so much for joining. Have a good night. I'll end the session for now. Thank you.